Welcome to Longevity by Design.
Hello, and welcome to another episode of Longevity by Design. I'm Ashley Reaver, and we are joined by Dr. Gil Blander. This episode is brought to you by Insight Tracker, a company that creates evidence-based health solutions that are simple, clear, and actionable. Today, our guest is Dr. Brian Kennedy. Dr. Kennedy is internationally recognized for his research in the basic biology of aging and as a visionary committed to translating research discoveries into new ways of delaying, detecting, and preventing human aging and associated diseases. He is a professor in the departments of Biochemistry and Physiology at National University Singapore and director of the Center for Healthy Aging in the National University Health System. Welcome.
Thank you. Thanks for inviting me.
So Brian and myself used to spend time at Leniger Rental Lab at MIT. Actually, Brian used to be at the lab much earlier than me because he was the first graduate student that worked on aging.
You can just say I'm old. It's okay.
Me too, but I spent time in different labs. And Brian is the first person that found STIR2 as a longevity-related gene. So it's very exciting to have Brian together with us. But my path with Brian crossed away a few years ago when we developed our product that called InnerAge, and I was looking for a statistician. And I was starting to send email to all the people that I know that work on longevity and try to see if anyone know. So I met Brian in 2015 when I was looking for a scientist to help me with the InnerAge product. And I was looking for a statistician. And Brian was kind enough to introduce me to one of his graduate students. His name is Mark, that helped me to develop the InnerAge product of Insight Tracker, and I really appreciate it, Brian. And I'm also really looking forward to hear more about your research and what you have done, and I'm sure that it will be a fascinating discussion. So looking forward.
Sounds good. I hope so too. You know, I always try to recommend my graduate students because they get tired of my ideas. So anybody else's idea sounds good to them. So.
Well, Brian, before we dive into your research, please tell us a little bit about you, where you currently live and work, since you are not just a random guy we found on the street.
Yeah, so I am currently at National University of Singapore, where I'm running actually, let's say, two programs at the moment. One is the one you mentioned, the Center for Healthy Longevity. And there we're really trying to take clinical approaches to demonstrate that interventions can have effects on human aging. And I'm happy to talk about that. And we've recently started a new program, the Asian Center for Reproductive Longevity and Equality, which is funded by Bioeco Foundation. That's Nicole Shanahan, Sergey Brin. And the idea there is to understand declining fertility in women and try to develop interventions that allow women to have more choices about when they want to have babies. And you know, the East Asia, particularly, the birth rate is exceptionally low, and one of the reasons for that is that women have to make tough choices between career and family. And if we can find ways to preserve fertility in a healthy way, that will allow women, if they want to, focus on career a little bit longer and then switch to family later. So we're also looking at postmenopausal changes there. So that's sort of one foot, and the other foot I have is in the private sector, and I'm involved with a number of biotech companies, and we're actually starting a geroscience investment fund here in Singapore, which I can't talk too much about because it's not fully approved yet, but hopefully soon we'll be helping to recruit the biotech sector around longevity into Singapore.
Excellent. So awesome. Brian, did you always knew that you wanted to be a scientist?
You know, I decided, yeah, sort of. My mother was a math teacher and my father was a chemistry teacher, and so it was sort of preordained, I think. But I read a book on, funny enough, on biotechnology and gene therapy in the 8th grade. And that's when I decided I wanted to be a molecular biologist. In retrospect, it's kind of a stupid way to choose a career because you really have no idea what you're selecting at the time, but it worked out okay. And the funny thing is, people are saying now, gene therapy is finally starting to work. So what is that? That's 40 years later. So it's maybe we'll actually get there with gene therapy. So.
That's cool. So can you give us a bit of a glimpse of what happened when you started your graduate study at Lenny's lab, and how have you got into the SIR2 family and found the protein? That's fascinating, at least for me.
Sure. When I went to Lenny's lab, I'd worked on yeast cells as an undergraduate. And so when I first went to MIT as a graduate student, I thought I'm going to do mice. And then I realized that it takes forever to do mouse experiments. And so now I have a lab full of people doing mouse experiments. So that's, I guess I shouldn't.
That's what I wanted to say, but
but I decided at the time I wanted to go back to yeast. And then, I met this other graduate student, Nick Austriaco, who's gone on to become a priest. Actually, that's another story. And we decided to work in Lenny's lab, but we thought, what's the craziest thing we could do with yeast cells? And we decided between apoptosis, which was a bit odd for a single-celled organism, and aging. We thought aging was less impractical, but it turns out that there's a whole field around yeast apoptosis now. And Nick, the priest, is a priest biologist, and he has a small lab at Providence College looking at apoptosis in yeast. So everything comes around, I guess. And so then we thought, how most of the yeast studies at the time had been correlative. First of all, it's very hard to do yeast lifespan studies because you have to. The yeast aging is the number of times a single cell can divide, so that it produces a daughter cell, and you have to remove it with a small needle. And so every 2 hours you go in and remove the daughter cell and keep a tabulation. And so it took a while to figure out how to do that in more of a, I'd say, a medium throughput way. And then we were thinking, let's just don't do correlative science. Let's try to find mutations that were longer lived. But we didn't know how to do that because you can't screen enough strains to find it that way. So we had a bunch of plates with tetrads where we dissected different strains of yeast and had them in the refrigerator. Really, we forgot about them. And then we thought a few months later, maybe we should do something with those strains. And we got them out, and we realized that the long-lived strains were all still alive, and the short-lived strains were died. So they were sensitive to putting them in the cold. And so then we could find mutants that were cold resistant, and then secondarily check to see if they live longer. And that's how we got to sirtuins. Now, the weird thing about that is that correlation only holds in that one yeast strain. If we'd done that anywhere else, that strategy wouldn't have worked. And I think that it's an important lesson for scientists is that whenever you listen to a professor talk about their work, it all sounds like they sound like Sherlock Holmes, and then A happened, and I thought, for a long time and deduced that B was going to happen. It never works that way. Serendipity plays a huge role. And that was a big break that we got, but we were also opportunistic. We took advantage of it. We went after it, and we generated a lot of papers from it. So it's I tell people all the time, if you get the weird result or the unexpected result, that's something that no one else in the world knows. And so follow it, because it may be really interesting.
Maybe that was the divine intervention that your colleague needed to become a priest. It just appeared.
I wouldn't know about that, but maybe.
Walk us through kind of what a day in the life looks like for you as a scientist in Singapore.
Well, these days, a day in my life is Zoom, 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 Zoom. But actually, the lab's actually up and running. Has been. We had a very short shutdown here for the pandemic, so we have a little outbreak right now. So we're all trying to be careful. But Singapore's done a great job handling the pandemic. I think that what I end up doing is trying to balance my time between administrative roles, running programs, my own lab, because I have a lot of people in the lab, and we also have a clinical component going now, and then trying to also facilitate getting the companies off the ground, because there's so much money now in the private sector, and the money in the public sector is still flowing slowly. So I feel like there's a lot of opportunity now working with companies, starting companies, working with investors. So, I really have a difficult time balancing things right now. And the challenge I have the most is that just sitting down and reading papers and keeping up with the literature, because it's like every minute of my day is filled with some kind of meeting. So, these are first world problems. I'm very fortunate to have the opportunity to do what I want in the laboratory and do the kind of research I like. So I'm not complaining, but it's a bit crowded right now in terms of time. So,
We call it the Zoom fatigue.
yeah, I know. And I, by the way, I don't think it's the China virus. I think the Zoom started the virus.
Zoom and Peloton.
Yeah, they've certainly benefited. All right, well, let's hop into some of your research. First kind of topic that I'd love to learn more from you about is caloric restriction and how caloric restriction and intermittent fasting can contribute to longevity.
Yeah, so calorie restriction was shown, I don't know, 80 or 90 years ago now, to extend lifespan in mice and rats, and it's a very consistent effect. It works in a lot of different mouse strains, not everything, of course. It works in all kinds of organisms, and at least looking at metabolic data, it would suggest that it would work in humans as well. So what do I mean by calorie restriction? I think what I would define it as the reducing calories down to a level that's just above malnutrition. And so you get everything you need, but nothing else. Now, the problem with that is that if you're a mouse and you eat pellets that we give you, we can control exactly what's in those pellets. But if you're a human and you're, eating like everybody does, it's hard to control that balance. And if you're not getting any excess calories, then you have to be exact about what you're getting and make sure you get all the right micronutrients. There are a lot of people that do calorie restriction that really think it works for them, but I think for the majority of the population, it's just very difficult to achieve. And that's one of the reasons that fasting became so prominent, because I think it's an easier thing to do. It's more sustainable for people. And that sort of came from the mouse studies originally, because what people realized is that when you have ad lib mice, so that, in other words, they have food in the cage all the time, they eat sporadically. They're active at night, so they eat mostly at night, but they may wake up in the day, eat some, then go back to sleep, and when you give the other group of mice 60% of that food, and there's 4 mice in the cage, they fight for the food and eat it right away, and then they don't get food for 12 hours, and then they eat the food right away again. And so even what we call the calorie restricted mice in most studies were getting fasted as well. And so people got the idea that if you do every other day feeding, which means you give the mice all the food they want one day and fast them the next, that might affect aging. And it turns out that those mice eat pretty much almost as much as control mice do, because they eat twice as much on the days they have food, but they still live longer. And so that was evidence that this fasting regime is really beneficial. And so now that's led to a lot of people. I know I'm not in a normal community in the academic space, but everybody I talk to these days is doing 16-8 or 12-12 or something.
Mm-hmm.
And there are companies now that are like El Nutra, which is produces diets that mimic fasting, and that was from the work of Walter Longo. So there's a lot. I'm really encouraged by that. I think it's one thing that people can do now that if they do a fasting approach is pretty sustainable, and it probably gives them a beneficial effect on aging.
Yeah, exactly, and it's actually very easy to do. It's not like you two decision when to start and when to stop, and that's it. So it's not not as hard as other diets that you need to make a lot of calculation. So Brian, my next question is about the sultanines, and what is the role that they are playing in aging?
Good question. When you find out, let me know.
Okay, I will call Lenny.
I think one of the challenges with all of these interventions that extend lifespan, and that's TOR inhibition, SIR2 activation, SIR1 activation, or SIR2 activation, and many of the others, AKG, spermidine, is that how do you determine what their mechanism of action really is that's linked to aging? So you probably talked about hallmarks or pillars of aging, these pathways that we look at that we think are important for the aging process: inflammation, macromolecular damage, DNA damage, proteostasis, metabolism, those kinds of things. The problem is that any intervention that extends lifespan improves all of these things. It improves proteostasis, it improves stem cell function, it improves all of this stuff. And so it's really hard to say that, well, I fixed DNA damage with this intervention and that's why the organism lives longer. The way I think about aging is a bit different. I think that really what we're looking at is a homeostatic network. So things are happening to you as you get older. You're getting damage to proteins and DNA and lipids and other things, but your body's compensating. You have this like cell signaling communication network that compensates for damage, keeps you functional, keeps you active, keeps you disease free. When you get enough damage, then you can't compensate anymore. You spiral out of control, and depending on your genetic risk factors and your lifestyle and other things, you start getting chronic diseases. Now, maybe that's diabetes, maybe it's heart disease, maybe it's Alzheimer's, or some combination thereof. So I think a lot of these interventions, what they really do is preserve that network function. So they probably act at nodes of the network somewhere so that they can add robustness to the signaling, and that gives you the benefit. And so you can read that out with all the down outputs, but that doesn't mean they're directly affecting all the outputs. And I think sirtuins are very similar. It's pretty clear that activating sirtuins are affecting aging, especially health span parameters. I particularly like SIRT6. I think that the data on SIRT6 is really robust. In that case, it may be through limiting senescence and preserving genome integrity. But I think that we could sit there and discuss why is TOR extending lifespan, why are sirtuins extending lifespan, and all of these things, and we really haven't figured out the answers to that yet. It's one of the things that's frustrating about the aging field. The good news is we're identifying all kinds of interventions, and we've got biomarkers now. But the problem is we haven't mechanistically done a great job of putting the whole puzzle together yet. And I think it may be that if it's really a systems level problem with the network, that's the issue, right? We're all used to solving problems one at a time, and really it's this integrated network that's controlling aging, and it's hard to think about that. And so that's my excuse anyway for now.
Okay. That's fair. And speaking on which, there are a lot of other molecules, as you mentioned, that have been related to longevity or aging. I heard you talking about alpha ketoglutarate. I know that metformin is very exciting, and a lot of even there are a few human trial about it, and NR and NMN and rapamycin and so on. So can you talk a bit about those and maybe mention which of them is your favorite today?
Whichever one makes me the most money. So. No, I hope I'm joking. So I think that the gold standard is still rapamycin, because it seems to work in every animal model. It's very robust in mouse studies. Multiple labs have got it to work in multiple strains. If I had to put a bet on something working, I think that rapamycin would be the biggest bet. The problem, of course, is that there are side effects with rapamycin. A lot of people are taking it for aging. They call me and tell me their results, but there is a small clinical study that I'm not involved with, I've heard of, that's starting now. I think it is possible to take it without side effects, but it's something to be a little bit careful with. But in animal models, it works great. My opinion, metformin, the animal data is not as robust. But there is some human data, like retrospective studies of people with diabetes, that look promising. And I'm very supportive of the TAME trial that Nir Barzilai is doing. I think that's certainly one way to address the health span problem. It's a very expensive way of doing it, because it's 14 centers and 3,000 people and tens of millions of dollars. But if you can prevent multiple diseases simultaneously with something that slows aging, that's a very big positive step forward for the field. And then, there's a lot of natural products, and I really like alpha-ketoglutarate. It's research that was done in my lab, and I'm involved with the company that's selling it, or one of them, which is Ponce de Leon Health, so full disclosure. But in mice, what it did was compress morbidity. So it extended lifespan by only about 10%. We started at middle-aged mice, but it compressed frailty by 50%, so or reduced frailty by 50%. So the animals stay very, very healthy and crash at the end. And there are clinical studies going on now with the company, and we'll probably test the product the company produces is not just AKG, it's AKG plus a vitamin, and they're doing clinical studies with that. And then over here in Singapore, we're going to do just the AKG and see if that works. I should also point out that the AKG that we're using is a sustained release version, so it's a lipidized version that gets through your gut and releases in the intestine. We think that a lot of the stuff out there that doesn't do that gets dissolved and doesn't have much of a bioavailability. So, but anyway, we're very hopeful for this. We have some clinical data already, but the study's not finished, and we also have some data from people that are just buying the product and doing methylation testing. And all of that is promising, but I want to see the clinical study done before I get too excited about it. But I'm pretty hopeful that it might work. And one other point about AKG is that people have taken it for exercise for a long time. Bodybuilders take it, athletes take it, and it's purported to increase endurance and improve like weightlifting and things like that. And so a lot of people that we notice that are taking it are reporting that their exercise is better. And so that's one thing to think about especially with supplements, is that, it's going to synergize with lifestyle. So if you feel better, you have less inflammation and you have more endurance, you're more likely to exercise, and then you get benefit from exercise. So, I think that ultimately what we're trying to do is find the right combination of things that really promote people toward healthy behaviors. And this may be part of the effect of AKG. NMN so far in our animal studies, we don't see big effects, but other people have reported health span benefits, and I'm waiting to see more clinical data on those as well. I think that there's certainly people I know that think it's the best thing ever. I have friends that like sprinkle NMN on food and everything else. So.
I assume that the same people that I know.
Probably. And but there's a lot of things coming down the path. Spermidine, at least in our hands in mice, that works. We haven't published. That'll be coming out soon. We just got the paper accepted, and then things like urolithin A, which is pretty interesting. So I'm very excited where the field is. I think you can point to like probably 50, if you look deep enough, 50 different things that may affect human aging, and I think none of us know for sure which ones are going to work best and which ones are going to work best in which people. And so that's what we want to do here in Singapore, is start to agnostically test interventions. We're going to use biomarkers instead of disease prevention, and try to see which ones have the biggest effect. And ultimately, if we combine them, can we do better? Those are the really simple questions, actually, but it's harder to do those studies than it looks.
So what you said at the end is exactly the beginning of my question. So it's very hard to do a longevity studies in human. You might need 100 years. So biomarker are keys. And recently the scientific community developed a lot of new aging-related biomarkers. So can you elaborate a bit about that?
Yeah, I think that many years ago, the National Institute of Aging realized the need for biomarkers, and they put a lot of money into studies to try to find biomarkers of aging. And then maybe 5 years after that, or 10 years, they concluded it was a failure. They didn't really find anything. And so, even in 2010, the best biomarkers were things like walking speed, time to get up and go, grip strength, maybe things like pulse width velocity, which do tell you something. Some of those only work well in people over 70, and so if you're doing interventions at a younger age, which is what we want to do to prevent disease, then you need other kinds of biomarkers. And Steve Horvath and Hannon and some of the other people really came through with these AI-based biomarkers looking at deep data sets, in this case DNA methylation patterns, and so they generated these methylation clocks. So the way they're done, or originally, were to take a person's chronologic age, so that is factored in, and then look at a large number of methylation changes in the genome, and let machine learning optimize those methylation changes to the age of the individual. And so if I'm 54, and which I am, and the biologic age says I'm 50, the argument is that that's not an error of the measurement. What that's really measuring is your biologic age, and so you're aging better than your chronologic age says. And we all know, if you look at 10, 50-year-olds, you don't need, a DNA sequencing machine to figure out that they're aging at different rates. We just need a way to measure. And so this methylation clock is, at least I think, I tend to believe that the data is telling us something about aging. So we're using those kinds of clocks. The one big question is, does it have to be DNA methylation? Because there are clocks that are made from transcriptomics, from microbiome composition, from we're using a clock that we reconstruct the three-dimensional view of your face, and we can predict your biologic age from that. Not your skin, but the age of your whole body we can predict from that. And then we're using things like inflammatory markers and metabolome. And so one of the questions we have here in Singapore is, first of all, we have ethnicities that have been understudied, like Malay and Indian populations. But second of all, how do these biomarkers of aging connect to each other? So if one of them says you're old, does the other one say you're old? Are they measuring the same things, different things? And what we want to do is to figure out where the overlaps are and where the uniqueness is, and ultimately come up with a $200 test that we can apply to everyone in Singapore that measures biologic age. And so that provides another kind of restriction too, is that it has to be affordable, because we can't spend $20,000 a person to measure age. And it also has to be minimally invasive. I mean, ideally not even a blood draw, but at most a blood draw. If we have to do a biopsy, forget it. I can't scale it. So that's really our limitations. And then, the first study, which is up and running right now, is to take people at different age ranges and measure all these biomarkers and get that data. The second study, which will start in a couple months, is to look at the first intervention. And we chose exercise first, just because it's something, we have exercise physiologists that know how to do this, and we wanted to have a successful first study to get, not cause any problems. And so, but the next thing we do after that is going to be supplements. So we're going to try the time-released AKG. We'll probably do some kind of NAD precursor. We're still trying to figure out what to do there, and then go on from there. And we want to test 10 or 15 different things and try to figure out really what works the best. Because I think everybody will tell you what works the best, but nobody really knows right now, because there's just not enough human data. So.
Yeah, I agree. It's might be a bit too early.
Yeah, but I do think some of these things will work. And I don't think it won't shock me if you get 5-year reduction in biologic age from some of these things. I think it's totally reasonable.
Yeah,
you can get that from exercise now.
I've seen a paper that published about the Horvath's clock a few weeks ago that actually show a very small study that showed that I think that was exercise and lifestyle that decreased the Horvath's age by a few years, and it was very small, but it's very encouraging.
Yeah, those kinds of studies are important, and we want to do them a little bit larger, a little bit more placebo-controlled. I'm not criticizing that study. It's very hard to get things done right now.
Yeah.
But we want to add a little bit more control to it, and really try to get data that we can go to the public sector and say, you guys, this is a cheap way to keep your population healthy 5 years longer. You should be able to do this. And I'm also excited by some of the more radical interventions. I mean, we're working with a company that does stem cells. And so I want to see whether some of the more expensive things work or not as well, because anything is scalable if it works well enough. And so we're kind of working at both ends. On the academic side, I'm more on the supplement and repurposing drug type approach because it's affordable. But on the private sector side, we're willing to try anything as long as it's safe.
So you mentioned a few things related to age so far. One study in middle-aged mice is but where you directed an intervention, as well as some of the other biomarkers like grip strength really only being meaningful in individuals over 70. Do you feel, or has your research found, that after a certain point it's too late to start implementing lifestyle modifications to slow the train once it's left the station of aging?
I think that's another fantastic question that we don't really know the answer to. The assumption is that there will be a point that you get limiting returns afterwards. That's kind of true with exercise, right? But the thing with exercise is that it becomes physically harder for frail people to do the exercise too. So that may not be a great example. But our clinical studies or intervention studies are going to be in people 45 to 65 because those are people at risk. We're choosing people that don't have multiple chronic diseases already. And we think that we can that's where we can get the biggest bang for the buck. But it's also the population that's really interested in anti-aging interventions right now. The older generation it's become ingrained in them that you can't do anything about aging. So it's much harder to get people in their 80s to do something. Now, I will say that we do have people buying the AKG product, and so they get a baseline methylation test, and they can do a follow-up methylation test. And this is not a clinically controlled study, but we do get the results back if they provide it. And we have seen people that are in their 80s that don't, by the questionnaires they fill out, don't seem that healthy, that have gotten a response. So it may be, I think the assumption is that once you get into multimorbidity and frailty, then it may not work. Things may not work, but I don't think we really know that yet. So it's an important thing to test. Right now, we want to work in the age range we're in because we think we can see an impact there. But it'll be critical to see how far you can push that out and still get benefits. And we really don't have any human data on that. So.
Kind of on that same vein, is there an age roughly when most people start the aging process?
Well, I think it starts at birth or preconception.
Sure.
Probably. The question, I think this gets to a bigger issue that we need to change medicine, and because so much of medicine, the first thing medical students learn is the Hippocratic Oath, right? And the way that gets in, which is a brilliant document, by the way. I don't want to criticize, Hippocrates, but it gets interpreted as first do no harm, and then it gets interpreted as don't do anything to healthy people because you're taking too much risk. And I would argue that nobody's completely healthy. That by taking a 40-year-old coming in the doctor's office and not giving them the attention and care that they need, you are doing harm. In my opinion, doing nothing is harming people. And we're so ineffective at preventative medicine, which is really what these aging interventions are, at least now, that we need to change that. We need a revolution that says, all right, we're doing sick care. We do it pretty well, actually. But we need to do healthcare as well, because that will reduce the number of sick people over the long term by probably a bigger margin. And it also will improve functionality, quality of life. And by the way, it's cheap. So, that's the revolution we need to create. And to me, healthcare is a lifelong process. It's not something you do after people get sick. So I'm not saying that I want to take a 20-year-old and put them on a bunch of anti-aging interventions, but I do think that you probably want to spend more time with younger people, trying to get them to adopt healthy lifestyles, healthy diets, And then, at some point you might transition to supplements and stuff that slow aging. And then at a later stage, you might be willing to take even more risk. But just leaving people alone until they hit 45 and then they come in with diabetes is not a good way to deal with the healthcare in the population.
Yeah, Brian, that's exactly the reason why I founded the Insta Tracker, because we have a sicker, and everyone that is not sick, basically nobody take care of him. So we are trying to be at least one of the solution for this problem. So I think that we are completely in line here. Thank you.
Yeah, no, I agree.
Awesome. All right, let's see. What emerging science could be helpful to better personalize interventions for healthy aging in the future? It sounds like that's something that your research lab is trying to specifically get at, who to assign which intervention, maybe combine them. Where do you see science going there?
Well, part of what we're doing is in mice there. And one thing we've learned, and I just want to, it's worth mentioning this, that there's a lot of products out there that you get a pill and it says it has 15 things in it. First of all, if it's this size, the AKG pill that we give is this size, and all it has is AKG and a vitamin in it, and we didn't make it that big because we wanted to. It had to be that big to get the ingredients in it. If it's this big and it has 15 things in it, you have to think about how much of each of those things is actually in that pill. But anyway, we've gone back and in mice, we've done a lot of combinations of different natural products. And the one thing I'll say is that I can't predict what's going to synergize. Sometimes we see things cancel each other out. Sometimes we see no big effect. And so I don't think we have much knowledge yet in terms of combining different interventions. And if I can't make reasonable predictions in a mouse for what works, then I don't think anybody can in a human at this point. The other thing is that it's not just which intervention for which person, it's how much. And so let me give you an example. We have a paper we're going to publish with vitamin A. And what we found is that with vitamin A, with the first vitamin A is a natural product, and everybody says, oh, that's safe. But it's actually not that safe. If you take too much vitamin A, it accumulates, it can cause toxicity. And so the first time we did the mouse study, we started 18 months. And we gave too much, and you're right. And we reconfirmed that it definitely causes toxicity. And then
it was an expensive way to confirm it, isn't it?
Yeah, expensive way to confirm. Yeah, and then we tried to lower. What I did is I did the same amount because we were seeing some potential benefits to frailty, even though the animals were dying. So I did the same amount for 4 months and stopped. And then what we found is that 20% of the mice still had the toxicity and died early. The other 80% lived 20% longer than controls. So now we're trying to use AI to optimize the amount of vitamin A for each animal. So we look at blood markers and try to look at toxicity and efficacy markers and try to optimize in each animal, because I think we can have a really big effect. And this is going back, it gets back to the vitamin question. Everybody says, oh, the meta-analysis shows vitamins don't work. How many of those studies did anybody actually measure the vitamin levels in the body of the people they were testing? Almost none. And so I certainly believe there's an optimal range for these vitamins, and that it's important. But unless you measure the individual, you don't know how much to give them of a supplemental of a vitamin. So I think we could take just vitamins, and if we did this right and controlled it properly, we could probably have a huge impact on aging. But it's going to take different levels in each person in terms of a supplement, and we have to figure out how to do that. So that's the first personalization type thing we're doing. And then, of course, as we start to accumulate human data with interventions, we'll hopefully be able to link efficacy for a particular intervention to traits of the people that are being tested. I would be not at all surprised if some interventions work in females and not males, or vice versa, because that's true in all the mouse studies. But beyond that, we might see that people that have certain traits like smoking don't respond to this, or that do respond to that. So but we have to have a lot of data to really start to draw those conclusions. And so hopefully we could, like, as more and more people are doing these kinds of study, we can find some way to compare each other's data, because we have to get the numbers beyond 50 per study if we're going to make those kinds of discoveries.
Yeah, so talking about the vitamin and the effect on human, I couldn't agree with you more. We have a database of tens of thousands of people, and we can see that in some people, 1,000 IU vitamin D will do the job, but in other you need 5,000 IU.
Yeah.
And we're starting to find why some need more and why some need less. So I completely agree with you that it's not only what, it's also how much. And I'm very excited about the experiment that you're doing about that. It should be very interesting to see the result.
Yeah, we're happy to compare notes too if you want. So
yeah, we'd love to. My next question is about the future of the longevity research and the aging research. So where do you see this field in 5 or 10 years?
It's funny. That question, is getting asked everywhere these days. There was just a New York Times thing on it, and recently, and there's a couple groups in Europe trying to put together, for political reasons, to prepare politicians for changes with big scientific discoveries. And I give everybody the same answer, is that I don't know. But I hope we're not where we are now. It's really, you've been in this field. It's been frustrating. I have this slide that, with the aging in the middle and arrows to all these different diseases. And Matt Kaberlein and I have used that slide for, I don't know, 15 years. And every time I see that slide, I want to just like go off the stage and have a shot of whiskey. It's like, we should make it into a drinking game, I guess. It really is. We've been trying to make this point for so long that aging is the biggest risk factor for everything, and we need to do something about it. It seemed like common sense to me at the beginning, and it seems like completely obvious to me now. But it's still been hard to convince politicians and clinicians and other people that this is true. The good news is there's a lot of private sector money right now. So investors have decided that there's something to be gained here. And that's allowing a lot of us to do research and develop interventions that actually work. So I'm very optimistic in 5 to 10 years we'll have good data that some of these interventions are actually having an impact on human aging. And hopefully we're thinking of ways to scale them into population at that point. I also think that things like mesenchymal stem cells, gene therapy, they show a lot of promise, and hopefully we'll be getting into the clinic and more meaningful human data to show that they impact aging. I mean, lots of people pay for MSC injections now, but the aging biomarkers haven't really been looked at in that context yet, for instance. So.
Awesome. So wrapping it up, I feel like you gave us a pretty good framework of approaching aging through the lifespan, kind of starting with lifestyle, maybe then getting slightly more aggressive with supplements, and then finishing with perhaps the riskier things as more research comes out of them. If there was, one thing that you could tell all of our listeners right now, maybe one decision that you intentionally make every single day based on longevity, what's something that you could share with our, the listeners that they could start doing right now?
It's basically, I'm going to sound like their mother, but there are 4 things that I think really impact aging. Sustainable exercise, some cardiovascular, combination of cardiovascular and resistance training. I'm very adamant about that. I do a lot of exercise. I do really long runs here in Singapore, and I'm not sure whether that's good for me or not in this jungle, but it gives me more energy for sure. Healthy diet, and we don't have time to go into that in too much detail. Maybe incorporating fasting, and then stress management, which I think the first big step is self-awareness, at least knowing when you're stressed, and sleep, which is not that easy to manage sometimes, but it's important. So, any little changes that can be made in healthier directions in those areas will have an impact. And not only will they have an impact 20 years from now, but they'll have an impact now. But they have to be sustainable. If I tell people to go be vegan marathon runners, they're not going to do it. So it's a matter of incorporating things into your lifestyle that you like and you can sustain. And if you can do that, you'll get benefits from it for sure.
Awesome. Thank you for sharing that and also for joining us today. It was really great to learn from you.
My pleasure. Happy to join anytime.
Thank you so much, Brian. We really appreciate it.
And we are looking forward to exploring more of the research in the field of longevity with you each week and the leading scientists here with Insight Tracker. Thanks so much.